Hello dear students, welcome back to Sajjan biology classes, and after a long time we are meeting. So today we are going to discuss that is a very important concept called an RNA interference. And RNA interference is nothing but a targeting a specific mRNA and where it is going to break down and do not synthesis a protein. So let us discuss that how actually the RNA interference mechanism it works. And here I have written that Meloidogyne incognitia, it is actually a nematode and which causes the infection to the roots of tobacco plant. And once after the infection, so it reduces the yield in a tobacco. So hence, next that novel strategy or novel discovery which has been introduced to prevent from this infection. So that mechanism is called an RNA interference. RNA interference is a cellular defense mechanism which takes place in all eukaryotic organisms including plants, animals, fungi and many. And let us discuss that how actually this RNA interference it works in plants to prevent to this infection. And the first thing is that the Agrobacterium tumefaciens vector and along with nematode specific gene, both are introduced into a plant cell. Once after the introducing into a plant cell, so they start to produce sense and antisense mRNA, okay, in plant cell. Once after forming the sense and antisense mRNA, both together become a double stranded, okay. And because of this double stranded DNA, so they can able to produce a double stranded RNA. And this double stranded RNA, once after the formation, there is a dicer. The dicer along with an RNA is 3 enzyme. So actually it is nothing but an riboendonuclease enzyme anta karitim. This riboendonuclease enzyme, what it will do, actually we are calling as an endonuclease. Hence it cuts the sequence of mRNA in between, not at the terminal part of the mRNA. Andre RNA madhyabhagadalli cut ಮಾಡುತ್ತೆ ಅಂದಂಗೆ ಆಯಿತು. Next ಏನಾಗುತ್ತೆ, after that, so by cleaving the mRNA in between, which produces the 3 types of RNA. So they are called siRNA, shRNA and miRNA. siRNA is nothing but small interfering RNA, and next the small hairpin RNA, and next microRNA. So any one of these 3 RNA, next it proceeds the RNAi. So let us discuss. When the infection it caused by the worm, so the any one of this type of RNA it enters, and where that RNA machinery it takes place inside the worm. So what it happens, along with these RNA there are certain protein complexes like in slicer and organode or the proteins, and it forms a RISC, you can see. So RISC is nothing but RNA inducing silencing complex. RISC is a multi-protein complex. It is a multi-protein complex. So what it will do, the RISC, it is a very important factor. Okay, RISC is a very important factor. So once after getting inside into the worm while actual infection, and which act as an unzipping factor. What does it mean of unzipping factor? So any one of the siRNA or shRNA or miRNA which enters into the worm, so it will be double stranded. So we need to make it a single stranded. Who will do that single strand mRNA? So that is nothing but a RISC factor. So what it will do, I told you that it is nothing but unzipping factor. So what it will do, it makes into a separate strands. And what actually the major function of this RISC factor is that, and once after the unzipping, so RISC factor it becomes activate and it is in the search of mRNA to make a complementary to that. Okay, so how actually it works, that RNA interference, I will discuss you. Until here, I hope you understood. Next, what it happens, so the casually within the worm, so where there is a DNA, okay, and DNA what it will do, so it undergo the transcription and produces the mRNA, okay, by the process of transcription, that mRNA it produces, and later what it happens, this mRNA undergo the translation in order to synthesis a particular required protein to that organism. And here we are targeting to this mRNA that should not synthesize a particular product of the protein. So how actually it stops the protein synthesis? I told you that where that risk factor when unzips the any one of the mRNA which has been entered. So I have taken an example, that siRNA, and this siRNA, what it happens? So it becomes unzipped, and now this single standard RNA is in the search of complementary mRNA, and where it releases the complementary mRNA, it becomes, means it finds the mRNA in the nematode and it becomes a complementary to this mRNA. When the mRNA it gets the complementary strand, it becomes a double stranded RNA. Once the mRNA strand, if it becomes double stranded, actually in the normal case of process, it will be in single stranded. But so already the risk factor, what it do? So it does identification of what? A single strand mRNA for the complementary purpose. So when that mRNA it becomes a complementary, so it becomes a double stranded. So hence the complete process of translation it stops and the worm it dies. This is what actually the RNA interference concept. I hope you understood, and still do you have any doubts about this concept, you can post or you drop your doubts or comments in the comment section. I will reply for your questions. Thank you. All the best.